Okay, hi everyone. This is our lecture on chapter six. I didn't have my coffee yet. Where we're sort of going to set up for and talk about, so a little bit about metabolism. So anyway, this chapter really sets up for the next two chapters on photosynthesis and cellular respiration, because we're going to talk about different types of reactions within those two processes. Okay. So it says energy and enzymes, right? So we'll talk a little bit about energy in general and enzymes in general. And again, this is all sort of applies to the next two chapters. All right. So in terms of the outline, right, we'll talk about just cells and the flow of energy sort of in general. And then we'll talk about metabolic reactions, okay, and energy transformations, and then some metabolic pathways and enzymes, and then oxidation and reduction reactions. So again, those oxidation and reduction reactions are reactions that are going to take place in photosynthesis and in cellular respiration. Okay. So in terms of just defining energy in general, right, we probably have heard of some of these terms before. So the energy is just the ability to do work, right, or bring about change. Okay, and if we're talking about kinetic energy versus potential energy, right? Kinetic energy is the energy of motion, right? Mechanical energy. Potential energy is stored energy, right? Or chemical energy. Okay, and so the cell is going to need to be able to tap into potential energy. I mean, I think we're going to talk more about this, but remember that energy currency of the cell is ATP, right? Adenosine triphosphate. In chapter 3, I talked a little bit about ATP, but basically there is stored energy within that molecule, right? Potential energy. And once one of those phosphate bonds are broken, then you release that energy, okay? And the cell can utilize that. So like I said, we'll talk more about that. All right, and so if you just look at this picture where it's just showing you the flow of energy, right? Sort of through an ecosystem here. You can see solar energy, right? And coming in, obviously, to the plant here, where the plant then can convert that solar energy into usable energy, right? And then make carbohydrates from that. If this animal eats the carbohydrates that the plant produces, right? It can then turn that into mechanical energy, right? And that's that energy of movement that it's using. And then you can always see that there is also always sort of energy that's released as heat. Okay, so energy is never really destroyed, doesn't go away, it just changes forms. All right, so that leads us to this. Okay, so you have to talk about the laws of thermodynamics, if again in biology, because these apply everywhere, right? So the first one is the law of conservation of energy. Okay, so energy cannot be created or destroyed, right? But it changes from one form or another. So I just sort of said that on that other, when I was just talking about that other slide, where you can see that heat that's lost. Okay. Oh, that's the only thing on that slide. All right. So this is continuing on with that same picture, right? So this is showing you, so what's happening in the tree, right, is photosynthesis. And I mentioned that, right? So if you just, again, look at that plant cell, because again, that's where photosynthesis is happening inside the cell, right? In the chloroplast. You could see, again, sunlight coming in, this plant cell being able to convert that sunlight into usable energy, make carbohydrates, right? Also needs to take in carbon dioxide, right, and water to do that. But then also some of that energy is going to be released as heat. Okay? If you look at the moose, the animal in that picture, again, I said this in the sort of overall picture, but they're going to eat that carbohydrate, right? They then can use that carbohydrate can be broken down, right, to generate ATP in order to let their muscles contract. But again, some of that energy will be lost as heat as well. Okay, so the second law of thermodynamics, right, is called the law of entropy. Okay, and so this has to do with disorganization. Now, every time I do this lecture, I sort of think about not really talking about this, but it's necessary to sort of understand. And so I'll go through it and hopefully it will be clear. Okay, so Entropy itself is just the amount or relative amount of disorganization in a system. Okay. And so obviously for our purposes in this course, we're going to think about it within the realm of a cell. Okay. Okay. So it says energy cannot be changed from one form to another without the loss of usable energy, right? Available to do work. So that's the idea of that heat being lost, right? Which we just showed in those last couple of slides. So that sort of waste energy, again, energy doesn't go away. It's not created, it's not destroyed. It just changes forms. That waste energy will actually go on to increase disorder. Again, increase disorder in some system, okay? So it says every energy transformation makes the universe less organized and more disordered and chaotic. Okay, so again, that sort of waste energy will create more disorder in a system. All right. So if we, again, like I said, our purposes, we want to think about this in respect to cells, right? So here we're going to look at kind of two examples. Okay, so this is showing you a glucose molecule, right? Glucose molecule that's intact, C6H12O6. And I hope just by looking at this versus looking at this, we would agree that this glucose molecule is more organized than this when it's broken down, right? So it says here, again, if we look here, it's more organized, it has more potential energy, it is less stable, right, in terms of entropy. So if we look at entropy is defined as the relative amount of disorganization. So this is really organized, right? So it has less entropy, okay? And and again, has a lot of potential energy. So the cell is going to break down glucose, right, to generate energy. Okay. And so here, and then it's essentially, like I said, generate energy. So it's going to release this kinetic energy. And so now you end up with this broken down glucose, right, in carbon dioxide and water. The cell was able to use the energy that was generated to perform all the other operations it needs to do. And now in this case, this is less organized. We all agree. Less potential energy. There is no stored energy anymore, right? The stored energy was here. That was already tapped into. And this has more entropy, right? It says it's more stable, but it's also more disorganized. Okay. So this molecule here, the glucose, it's sort of inevitable that it's going to be broken down, right? So that's why it's saying it's less stable. So it's more organized, but less stable. What's going to happen to it is going to be broken down. These molecules here are not going to be broken down further. So in that state, they are more stable. Okay, so now if we look here at this example down here, we see basically a membrane, right? A channel protein and some hydrogen ions here. And so this is showing you unequal distribution of hydrogen ions, right? You have a lot of hydrogen ions here. This is actually good for review from the last chapter too. By the way, make sure you have your exam 2 review sheet and are writing notes in there as you're going through this lecture. I forgot to, in chapter 5, I only reminded you the last slide, so I remembered a little bit earlier this time. So this is a good review from that too, right? So what do you have here? So let's kind of review first. You have a concentration gradient, right? High concentration of hydrogen ions here, low concentration here. If we talk about diffusion and the movement of these ions, right, they're going to have the tendency to go this way and they're going to move through that channel protein, okay? So again, here, this is more organized, right, but has more potential energy because these are going to have the tendency to move along their concentration gradient, okay? And again, less entropy, right? More organized, less entropy. All right. So what's going to happen in this system is that these hydrogen ions are going to move, right, towards the low concentration and they're going to try to reach equilibrium. Okay. So again, in this case, you have less organization, less potential energy, and again, it's more stable. And there's more entropy, right? It's more disorganized. Okay, so the idea in biology, if we're thinking about this, is that again, entropy refers to disorganization, and that things like a large glucose molecule or another large molecule, they're going to have the tendency to be broken down. That's what's going to happen in that system, and so therefore, the large glucose molecule is less stable, right? It does not have as much entropy because it's more organized, but it's going to eventually be broken down and contain and end up being more disorganized, right? And more stable. I guess the one analogy I guess that I usually use is if you think about like your dorm room or your bedroom, right? When it's super clean and neat, right? It took you a lot of energy to get that, right? You put all that energy in to get that room nice and clean, right? So it has all this sort of stored energy that you use to make it nice and clean. What happens though over time, as you're not really putting energy in, right? It starts getting messy, okay? And so you sort of, it's less organized, and basically at that point it's sort of more stable, right? That nice, neat, clean room inevitably is going to start to get messy. That's sort of my analogy. So anyway, I just want you to sort of understand the concept again as it relates here on this slide to this cell. It's nothing that I'm going to like ask you tons and tons of questions about, but I might on the exam include a picture like this and say, you know, which of these, this glucose molecule or it broken down in carbon dioxide and water, right, has more entropy, right? Or which one is more organized. But I may, you know, ask you in terms of entropy so that I know that you understand entropy. Now, I don't need to give this exact same example. I can give you a different example, right, to make sure we understand. So again, I could use a different molecule. I don't know, starch or something like that, or I don't know, some other larger molecule. And basically in its intact state as that large molecule, right, it's going to have less entropy, right? It's more organized. And then when it's broken down, it's going to have more entropy. It's more disorganized. Okay, so that's kind of what I just want you guys to understand there. All right, so we move on to the next section here, okay, to talk a little bit more about reactions that are going to occur in the cell, okay? So the term metabolism, again, for our purposes, we're going to think of metabolism as being all the chemical reactions that occur in a cell, okay? So if I asked you to define metabolism, that's what you would tell me. It's a sum of all the chemical reactions that occur in a cell. And there are lots and lots and lots of chemical reactions that are occurring in all your cells all the time, okay? Reactants participate in a reaction, and then products form as a result. Okay, we probably are familiar with those two terms. In terms of some of these energy terms here that relate to these reactions, so free energy, or delta G, right, is the amount of energy that's available to perform work. Okay, so we can talk about two different types of reactions here. Reactions that have less free energy than the reactants. Products have less free energy than the reactants, and these are reactions that release energy, right? And so exit, right? Release, exergonic reactions release energy, okay? So those are reactions that create energy, release energy. So think about an exergonic reaction as that on that last picture, breaking down glucose. That's going to release energy, okay? So again, the products have less free energy than the reactants. I'll show you another slide that clarifies this a little bit more. Endergonic reactions, on the other hand, the products have more free energy than the reactants. And so these require energy input in order for them to occur, okay? So again, think about synthesizing a molecule, okay? Remember we talked about the dehydration reactions and the hydrolysis reactions in general, right? Those reactions that are breaking down a molecule is going to liberate energy. If we're talking about synthesizing a molecule, that is going to require energy to be put in. So we just kind of talked about those two different types of reactions, right? In general, the dehydration and the hydrolysis reactions, but we didn't really talk about how much energy is required or what energy comes out. And so now this is talking a little bit about that. Again, we're not going to get into too crazy of detail, but we want to understand that. And also understand that those reactions require enzymes as well. And that's what we're going to talk about in a couple more slides. Okay. So we kind of in chapter 3 just talked about these different types of reactions. Know that those different types of reactions require energy or give off energy, because energy is always involved in any reaction that's going to occur in the cell. Okay. And that those reactions also require enzymes as well. All right. So, right, I mentioned this again, I mean, I mentioned this in chapter 3. I mentioned this at the beginning of the lecture, and I told you we'd talk about it a little bit more. So ATP is what the cell uses for energy, okay? It is used to drive metabolic reactions. Again, what are metabolic reactions? They're just any reaction that occurs in the cell, okay? So basically, you constantly have turnover from ATP to ADP. If you again go back to the end of chapter 3 where we sort of described that, right? Adenosine triphosphate, you cleave a phosphate bond off, you end up with diphosphate, only 2 phosphates, okay? So we did talk about already what it's composed of. And what happens, and I kind of mentioned this, I think, when I talked about that briefly, is that you can couple the breakdown of ATP to other reactions in the cell. That's how it works, right? So that the cell can utilize the ATP, utilize the energy, right? That's released from the breakdown of ATP to drive an endergonic reaction, right? So it says here, energy released by an exergonic reaction, right? Like ATP being converted to ADP can then drive an endergonic reaction. Okay, so remember, exergonic reactions release energy, endergonic reactions consume energy or require energy. Okay, so this is showing you the ATP cycle, meaning ATP going from ADP, and then it has to go back to ATP if it's going to be used again. Okay, so if I ask you about the ATP cycle, this is what I'm referring to. Okay, so here, and we're starting off with ATP, right? Note what it says: ATP is unstable. Just like that large glucose molecule was unstable, right? And has high potential energy, lots of stored energy in these phosphate bonds. All right, so if we talk about this exergonic reaction, like I was just mentioning, which is the breakdown of ATP, right? I think there's okay, which is the breakdown of ATP, right, into ADP plus a free phosphate. So basically we cleaved off one of the phosphates here, right? Okay, so here the hydrolysis of ATP releases previously stored energy in this phosphate bond, okay? And then now you can use that energy to drive other reactions, okay? It says it has a negative delta G. If you go back when we talked about the free energy thing, right? If the reactants have less free energy, right? We go back here. Products, so if we're talking about exergonic, right? Products have less free energy than the reactants, right? So the reactants have, it's going to be releasing energy. So if the products have less free energy than the reactants, you end up with a negative delta G, okay? Again, I'm not going to ask you too much about delta Gs, but I want you to understand that delta G means free energy. And then if you're talking about an exergonic reaction, it's a negative delta G. But I guess I more just want you to understand that an exergonic reaction is a reaction that's going to release energy, okay? Create energy, it's not really create energy, release energy, right? Because it's not created. That energy was stored, right, in these phosphate bonds here. When that bond is broken, it releases energy, okay? All right, so it says examples: protein synthesis, nerve conduction, muscle contraction. All right, those are examples of things that require energy, okay? Oops, wrong way. All right, so now once ATP is broken down to ADP, it's important that the cell is able to then convert that ADP back to ATP so that it can be used again to drive other reactions, right? So hopefully we understand that. And so if you going from ADP to ATP, so now you only had 2 phosphates, but now you added another one on. So it's if we think about, again, chapter 3, when we're talking about synthesizing molecules or breaking down, right? If you synthesize a molecule or you build something, it requires energy to be put in. If you're just breaking something and destroying it, it does not require much energy, okay? So here, this is endergonic, okay? So it requires input of energy from other sources. So there has to be some sort of input of energy. Again, has a positive delta G, and it says, example is cellular respiration, because in cellular respiration, the cell is making ATP that can then be used later on, okay? All right, So if you want to kind of illustrate this sort of coupled reactions, right? So when we say coupled reactions, we mean reactions that require energy, endergonic reactions, are coupled with the breakdown of ATP, okay? Because the breakdown of ATP, which is an exergonic reaction, releases energy, right? So that's what this is saying. So this is saying use of ATP during muscle contraction. All right, so here's just an example here, and it's not really, it's kind of showing you it on a bigger picture, okay? So it says here, resting shape when it combines with ATP. So this is just showing you this, this is myosin and this is actin, right? So remember, actin and myosin interact with each other during muscle contraction, okay? And the interaction and the muscle contraction is dependent, but it requires ATP, okay? So you can see ATP bound to that myosin there. All right, so now, now that ATP is converted to ADP, right? And energy is liberated, okay? And so that allows myosin, or causes myosin to change shape and allows it to attach to actin. So if you didn't have ATP available, right, the myosin would never be able to grab onto the actin, all right? Okay, so then what happens further down the line, then this ADP is actually released and it allows this myosin to again change shape and basically pull on the actin. This is supposed to be against and then a space, actin. And basically that generates force or motion. That's basically causing these two filaments, the actin and myosin, to slide past each other, okay? And it ultimately causes contraction of the muscle, as you can see here, right? She's doing a bicep curl. Okay, so in the relaxed state and then in the contracted state. So muscle contraction, movement of your limbs is dependent on ATP. All right, so we move on. Like I said, all of these reactions also require enzymes, okay? Yes, they require energy, a lot of them, or they liberate energy, but they also require enzymes, okay? So let's talk a little bit more about enzymes. So one thing about reactions in the cell, or, think about cellular metabolism or metabolism, right, means it's the sum of all the reactions that occur in the cell. Well, a lot of them occur in sequence. They usually just don't occur in sort of as independent reactions. A lot of them are sort of sequenced together, okay? So it says products of an earlier reaction become reactants of a later reaction, okay? And so these reactions are linked and they form something called a metabolic pathway, all right? And so this is illustrated down here. And so again, it says here, sort of reiterating again, begins with a particular reactant. So here's A, the initial reactant, right? And there's several reactions that occur before the end product is produced, right? So it's not like A and then just one reaction gets you to G, okay? There are 1, 2, 3, 4, 5, 6 different reactions in this metabolic pathway going from A to G, okay? So it says here, B, C, D, E, and F are all intermediates. We will call them intermediates. Okay, by the way, you need to make sure you guys understand this because I think this is a really important concept in biology. And so this is something that I might ask you in an open-ended question, right? I might have a metabolic pathway like this up or a different one and just ask you about it, right? What's the reactant? What's the product? How many different intermediates are there? Okay, so if you really understand what a metabolic pathway is, you'll be able to answer those questions. Me just asking you define a metabolic pathway doesn't really tell me if you understand the concept or not. Okay, so let's move on from there, okay, because there's more, okay? If we look at that same metabolic pathway down here, okay, you can see now we added something, right? So this is enzyme 1, enzyme 2, enzyme 3. Each reaction within this metabolic pathway is catalyzed by a specific enzyme, okay? Again, super important, right? In chapter 3, we were just talking about reactions, some chemical reactions in general. We didn't mention energy. We didn't mention enzymes, okay? It is necessary to have an enzyme for every single reaction that occurs in the cell, okay? So in fact, this with the enzymes in the metabolic pathway is more realistic, right? And more important even to understand, okay? So we still should know that this is the initial reactant, right? And that G is the final product, right? These are all still intermediates, but you have specific enzymes catalyzing each reaction. So I could say, what enzyme, right, catalyzes the reaction from going from C to D, and it's enzyme 3, okay? Let's make sure I mentioned everything that's written here. So it's a little bit more, actually, I didn't mention any of this. Defining what an enzyme is, right? So super important. So protein molecules that function as catalysts, okay? So almost all enzymes are proteins, but not all enzymes, okay? There are examples of enzymes that are made up of RNA, and they're called ribozymes. But for the most part, we think, most enzymes are proteins, okay? The reactants of an enzymatically catalyzed reaction are called substrates, right? So instead of calling A a reactant, we would call A a substrate, okay? Each enzyme accelerates a specific reaction, okay? So E1 cannot catalyze this reaction here. It is only specific for this reaction. And remember, we're going to have a lab, an enzyme lab, that actually then reiterates the sort of this point about enzymes, and also will investigate kind of what types of factors will affect enzyme activity. Okay, so we will have a lab that will help sort of emphasize this information a little bit more. Okay, each reaction, right, in a metabolic pathway requires a unique and specific enzyme. Like I said, these are specific. They're not just general enzymes that can be used for anything. So important that you understand that. The end product will not be formed unless all enzymes in the pathway are present and functional. So therefore, if you have some genetic disorder where you're just missing enzyme 3 in this, that's part of this metabolic pathway, it doesn't matter. G will not be made. And if G is a super important hormone, or something like that, your body will not be producing that hormone. Okay. So, super important that we understand this. Now there's something else that we're going to talk about that's tied to this. Once we talk a little bit more about enzymes, just to know. So we'll come back to this again, okay? In terms of how the cell sort of regulates these pathways, all right? So metabolic pathway, focusing now on enzymes, okay? So let's talk a little bit more about these enzymes. Okay, so in terms of the interaction of the enzyme itself with the substrate, right? The substrate binds to the active site of an enzyme, okay? That's where the substrate binds, that's what we call it. So we think about the enzyme as a protein, right? Most enzymes are proteins. There is a specific part of that enzyme called the active site that substrate will bind, okay? And so when that substrate binds, it causes the active site to change shape. Okay, it says the shape change forces substrates together and initiates the bond. So if you think about a synthesis reaction, right? So think about when you're joining 2 amino acids together to start forming a protein, right? Those 2 amino acids will bind to that active site, and basically now that enzyme brought those 2 amino acids together, close together, so that bond can form, okay? So we'll talk a little bit more about that. This is called, so there's something called the induced fit model in terms of describing the interaction between the active site and the substrate, right? So it says the enzyme is induced to undergo a slight alteration to achieve optimum fit for substrate. So once the substrate sort of initially binds, the enzyme will go through a slight change in conformation for that substrate to fit in there perfectly. So I thought I had a better picture of that, but I guess not. I'll reiterate that again. So let me go back to that. So if you kind of think about, let's think about this here, right? So this product, this is the substrate. Sorry, these are the substrates here. So let's go with this one because this is degradation. So here's the substrate, right? This one molecule here. Here's the enzyme. Here's the active site. So when this substrate initially binds here to the active site, the enzyme will actually undergo a slight conformational change, right? A change in shape so that this substrate exactly fits into that active site. So that's what that induced fit says. All right, so let me go back just to talk a little bit more about these reactions. And I've sort of been talking about them all along, right? Remember, we talked about degradation and synthesis reactions, okay? In degradation, right, enzyme complexes, this is what I was just showing you, will bind with a single substrate, and then that substrate gets broken down. If we talk about synthesis, 2 substrate molecules, I also mentioned this when I gave the example before with the amino acids in the protein, right? two substrate molecules, and then the substrates get joined together and released as a single product. So if we look at these diagrams here, again, we should understand these. So if I showed you, one of these pictures versus another, we should know which one is showing degradation and which one is showing synthesis. I also could ask you, tell me what this is. You should know this is the enzyme substrate complex. We should know this is the enzyme. These are the products. This is the substrate, okay? You follow the arrow. So here's your substrate, right? Binds to the enzyme, complex is formed, then that reaction occurs. And in this case, it's going to be breaking that bond, right? And then you have these two products released. So again, think of this as breakdown of a larger molecule, okay? And so we call that degradation. On the flip side, if we're talking about synthesis, right? Two substrates will join into that enzyme, right? You have that enzyme substrate complex, and basically these two substrates get joined together and it's released as a single product, and that enzyme is then released. By the way, the enzyme is not changed by the reaction, and it therefore then can go and catalyze some more of this specific reaction, okay? All right, so talking again more about enzymes here. It says molecules frequently do not react with one another unless they are activated in some way, okay? Energy must be added to at least one reactant to initiate the reaction. So we call this energy of activation. So this is the energy that's necessary for a certain reaction to happen, okay? So it says the energy that must be added to cause molecules to interact with another. And this energy of activation is lowered by the enzyme, right? Think about this. It says energy that must be added to cause molecules to interact with one another. So if you kind of think about this example here, well, the enzyme is basically lowering that amount of energy because it's bringing those two molecules together, right? If that enzyme was never there, it would require a lot more energy to get those two molecules to come together. Okay, so I said this already. Enzymes operate by lowering the energy of activation, okay? I just said this as well. Okay, so if we look at this graph, again, this is also something that you should be familiar with and really understand. And so if I took out these labels and asked you to then label them or tell me what things are on here, we should understand that. So first off, let's see what this is graphing. We have free energy here and the progress of the reaction here. The blue curve is where there is no enzyme, and the yellow curve is where there is an enzyme, okay? And so basically this peak here we call the energy of activation, right? The amount of energy that's needed basically to go from reactants to products. And so you can see in the blue curve, there's a lot more energy needed, right? But when the enzyme is present, it has lowered that energy of activation, okay? So again, you can see this energy of activation is kind of from here, from the peak to where those reactants started from, okay? Right? That's essentially what that is. Here, it's from, again, the peak and sort of where these reactants started out as, okay? So that's the energy of activation, much lower in the enzymatic reaction, okay? So again, within the cell, if you think about these reactions, if there were no enzymes, maybe some of these reactions might still occur, but they would never occur to the level or at the speed that's necessary for a cell to survive, okay? So again, the enzymes are there to lower the energy of activation, make that reaction occur essentially at a speed that's sufficient for the function of the cell. Okay, so yes, make sure you understand this entire graph and exactly what it is showing. Okay, now we'll talk a little bit, and again, this is what the enzyme lab will address. What are some factors that will affect the activity of an enzyme, right? Or the speed of an enzyme. So one is how much substrate is there. Okay, so it says enzyme activity will increase with substrate concentration. So meaning if there is more substrate there, you will have an increase in enzyme activity because there is more of a chance of the enzyme coming in contact with the substrate because there's more substrate around. Okay, so that's what this is saying, more frequent collisions between the substrate molecules and the enzyme. Okay, So there you go. Super simple concept to understand, I think. Another thing that will affect enzyme activity or enzymatic speed is temperature, okay? Enzyme activity increases when temperature increases. Why? Because molecules will move around faster when the temperatures are warmer, okay? And so there'll be more effective collisions between the enzyme and the substrate. However, if the temperature gets too high, the enzyme will denature, okay? So again, most enzymes are proteins. If they're heated to too high of a temperature, what happens is they lose their shape, they denature. Remember, proteins can only function if they are folded into their proper shape. If temperature is too high and they denature, they lose that shape, okay? So think about enzymes and substrates, right? The substrate will bind to the active site of the enzyme. Well, if that enzyme is heated and denatured, there will be no active site for that substrate to bind, right? So super important that temperatures are not increased too high. So that's like if your body temperature gets too high, right? It causes proteins to denature in your body, okay? Which then can lead to basically, organ failure and death if high enough for a long enough time. All right, pH. Okay, most enzymes are optimized to a particular pH. So meaning so it's not like if the pH gets too high, it's not going to work, just a kind of a general thing. There's a particular pH that each enzyme will work most efficiently at, okay? And that will vary depending on the enzyme and where they exist, all right? So let's kind of look at some of these things. So this is showing you the effective temperature on rate of reaction, okay? So one thing to think about, if you think about ectothermic animals, right, or cold-blooded animals, they don't have means to regulate their own internal body temperature. And so a lot of times the reactions are limited, okay? Essentially the rates of reactions are limited by temperature, right? So that's something that they really have to consider. If we talk about endothermic animals, right? Like this polar bear here, also ourselves, we don't have to worry about that, okay? We have our internal temperature needs to be maintained at 37 degrees Celsius, right? Because that's the optimal temperature for our reactions, okay? So this is basically showing you here, if you look at temperature, right? As it increases, the reaction rate gets higher. However, you get to a certain point where that reaction rate will drop, right? And why? Because at that point, those enzymes they start to denature, okay? And then once they're denatured, no function at all. So you can see kind of just past 60 degrees, basically the enzyme has been denatured, okay? So when you do your experiment, or when I do the experiment and do the demo, or when we watch the video, if we illustrate, effective temperature on the reaction and you draw a graph, you'll probably see a graph just pretty similar to that, okay? Where you have sort of the rate of reaction increasing and then it drops off. Okay, effective pH, this is what I was just telling you in terms of each enzyme has an optimal pH that it works best at. And so this is giving you two examples here. So pepsin is an enzyme that lives in the stomach, okay? So in the stomach, the pH is really low, like around a pH of 2, right? Because of the acid that's there. And so pepsin works optimally at a pH of 2, okay? As opposed to trypsin, which is also another type of enzyme that breaks down proteins. So both of these enzymes break down proteins. So think about them being involved in digestion, right? Food gets into the stomach, starts being broken down. Trypsin, on the other hand, is released by the pancreas and basically will work in the small intestine. It works at an optimal pH of about, between 7 and 8, okay? Because that's what the pH is there in that part of the body. All right, so the point is this is illustrating the point that each enzyme has a specific pH that it works best at, right? So this is showing you an example here of this cat, right? And so the color change has to do with the effective temperature on certain enzymes, okay? So these are Siamese cats, right? A Siamese cat, and they have an inherited mutation that basically causes an enzyme to only be active at cooler body temperatures, right? So only certain regions of the body are dark in color. Okay, so these are areas where the temperatures are cooler, they're dark in temperature. Where they're warmer, it's lighter. Okay, so again, it's just this inherited genetic trait that they have, but you can actually see it, right, by on the color of their fur. Okay, so anyway, that's just another illustration of the effective temperature on an enzyme. Okay. Okay, so some more, a little bit more about enzymes here. So it says cells can regulate the presence or absence of an enzyme of an enzyme. Cells can regulate the concentration of an enzyme, right? Meaning that depending on what metabolically needs to happen, right, based on cellular needs, their cells need to regulate the enzyme, right, in terms of how much is there and whatnot. There are also other ways that the cell can regulate the amount of enzyme or regulate the activity of enzymes, and that's by activating or deactivating them. Again, you're not going to have all enzymes on all the time. Cells aren't going to make things that they don't need, right? Because that would be a waste of energy. Also, having too much of something can end up being toxic to the cell, right? So there is a great need for the cell to be able to regulate enzymes, right? Regulate the amount of enzyme, like it says here, but also regulate the activity of enzymes, okay? So some ways in which the cell can regulate the activity of enzymes is something called a cofactor, okay? So these are molecules that are required to activate enzymes, okay? So just in general, right? Cofactors are molecules that are required to activate an enzyme, all right? So there are some examples here. So coenzymes are non-protein organic molecules. So whatever, these are still molecules that will be required to activate the enzyme. So it's not only the substrate that will basically activate that enzyme, but something else has to be there before that enzyme can function, okay? And those are called cofactors. Vitamins are actually components of coenzymes, right? So if you're deficient in a certain vitamin, your enzymes are not going to function properly because they contain these coenzymes, okay? Or yeah. Whatever, yeah. Think about vitamins as being coenzymes, okay? Okay, so it says they're small organic molecules that are required in the diet for synthesis of coenzymes. Again, if you don't have those vitamins, these coenzymes will not be made, and then that whatever enzyme that coenzyme works with will not be able to function, okay? So it's not just the enzyme and the substrate. There are other molecules that will regulate the enzyme and that are necessary for that enzyme to function. Okay, so an example here is vitamin B3 or niacin, right? Results in a skin disease called pellagra, right? So there are many other examples of diseases associated with vitamin deficiencies as well. And again, it's because then enzymes are not able to function properly, okay? All right, so this is showing you an example here in terms of what's happening on the enzyme, on the active site, right? So there is an active site here. The cofactor has to bind to the active site first before the substrate can bind, right? So if the cofactor is not there, this substrate will not be able to bind, and therefore then the enzyme's not going to function, okay? So cofactor just as important really as the enzyme in this case, okay? All right, so here's just another thing to talk about, and we're sort of kind of going back to that metabolic pathway here. In terms of something called inhibition of enzymes, right? So it says a substance known as an inhibitor will bind to an enzyme and decrease its activity. So with the cofactor example, we were really talking about increasing the activity, right? Or in terms of it being necessary for the enzyme to function, right? Here now we're talking about examples of ways that the cell can inhibit the enzyme, okay? Again, because if there's enough of a certain product, the cell doesn't want to make any more of it, so it needs to be able to inhibit, okay? So there's two different types of inhibition, okay? Competitive inhibition and non-competitive inhibition, right? Okay, so as I was just saying, so enzyme inhibition, again, is a way for the cell to turn off an enzyme, right? Or inhibit, slow down. Not really slow down, I would say think of it as kind of turning off its activity, right? But just for a given amount of time. We're not gonna, in most cases, we're not just gonna be inhibiting it forever, right? It's just for a certain time, and then it can be reversed, okay? So competitive versus non-competitive. So here, for competitive, the substrate and the inhibitor both bind to the active site, okay? In non-competitive inhibition, the inhibitor will bind to another site, not the active site, and we call that an allosteric site, okay? So let's look at, hopefully we got it. Okay, so let's look at a picture here. So again, like I said, this goes back to that metabolic pathway, right? I said we were going to come back to this. And again, this is again something we need to understand. So here, right, here's a metabolic pathway, right? F is the end product. We're starting with A. You have an enzyme that catalyzes each one of these reactants here, okay? And what do you notice that's happening here? So again, F is the end product. So think about this as a hormone being made. So think of it as being testosterone, okay? So this is the metabolic pathway. So you start with cholesterol, right? Have a series of reactions that, ends in the production of testosterone, okay? There is only a certain level of testosterone that's necessary and should be made within the cell, right? And so once that level is reached, the cell is going to want to inhibit the production of it. It does not want to make any more, okay? And so what it can do is, again, this F, the end product, can act as an inhibitor, okay? So what this is showing you here, so if you look up here, right, this is showing you the enzyme, okay? And this is showing you the substrate here, right? Enzyme 1, substrate A, binding to the active site, okay? So that's essentially, this is the active site. This is then going to cause this to turn into B, right? And move through the metabolic pathway. Again, however, once the cell makes enough of this end product, F, can act as an inhibitor, okay? And so if you look at this here, is this a competitive, right? Is this competitive inhibition or non-competitive inhibition? Okay? It's what? It's non-competitive because you see here, this is binding to a different site called an allosteric site, which is labeled up here. Once this binds to this allosteric site, notice what happened to the active site. It causes the enzyme to change shape or conformation so that now the active site changes and this substrate A cannot bind, okay? And therefore it inhibits this entire pathway. No more F will be made because the cell does not want to make more F, okay? Then once these levels get lower and the cell needs to make more, this will not be binding to the enzyme and it can then bind that substrate again, okay? Like I said, it's reversible, okay? All right, so that's what this is saying here. I just said this, right? A can no longer bind and it's inhibited. Okay, so really important to understand this process. I most likely will ask you about this on the exam. And again, I'll probably give you this figure and ask you specific questions about it, okay? I won't just say, what's an allosteric site? Oh, it's a site, separate from the active site. Great, but that doesn't mean you understand it, okay? So understand what's going on here. Again, if this is something I need to go over, please let me know and I can go over it again. One other thing I want to point out, or a question to think about, well, why do you think this end product inhibits enzyme 1? Why doesn't it just inhibit enzyme 5? Okay, so think about that. Why does it make sense for the cell to inhibit the enzyme that catalyzes the first reaction in the pathway rather than the last reaction in the pathway? Okay, so think about it for a second. And the answer is that the cell, if it just inhibited this one, well, then it wasted all of this energy to make all of these other intermediates when it didn't need to, right? So it makes sense to inhibit the earliest pathway there, all right? Okay, and also some of these intermediates may feed off into other reactions as well, and so things are a little more complex than it seems, but just think of it as it would want to inhibit the earliest reaction that it could, okay? All right, so finally, the last part of this chapter talks about oxidation reduction reactions, and this is something you'll talk about or go over in chemistry as well. We call them redox reactions, okay? And again, these types of reactions happen in photosynthesis and cellular respiration, which is the next two topics. And so that's why we're sort of introducing them here. So it will come back again a little bit in the next two chapters as well. All right, so what is a redox reaction? It's basically the movement of electrons, okay, from a molecule to another. I think it's sort of easier to think about the movement of hydrogens, okay, and it's easier to see that. All right, so anyway. Oxidation is the loss of an electron or the loss of a hydrogen ion, okay? Reduction is the gain of an electron or the gain of hydrogen, okay? So let's think of it as a loss of hydrogen or gain of hydrogen, okay? There is it's down here. There is a mnemonic for this, right? Oil rig, oxidation is loss, reduction is gain, okay? Now, if you use the example here, right, sodium chloride, you can't follow the hydrogens, but if we're talking about biology, right, in organic molecules, we usually are going to be able to follow these hydrogens, okay? And so that's the easiest to sort of see what's going on. So here it's saying sodium is oxidized and chlorine is reduced, okay, because we're talking about the gaining and loss of the electron. But again, I don't really like that example. We're going to give different examples here where we follow the hydrogens, okay? You always have these both taking place at the same time, right? So again, it's saying in the production of sodium chloride, sodium is oxidized, chlorine is reduced, okay? They always are happening together, okay? Okay, so one molecule accepts the electron given up by the other, okay? Again, or one molecule accepts the hydrogen given up by the other, all right? So I added these in here in terms of the overall reaction for photosynthesis and cellular respiration, because this is what we're talking about in the next two chapters. So if we talk about redox, right, oxidation and reduction, okay, let's look at carbon dioxide here, okay? Or we can look at these two molecules here, and we want to say which one is oxidized as we move over here and which one is reduced, okay? So oxidation is loss, right? Oxidation is loss, meaning loss of hydrogens. Okay, so if we look at water going to oxygen, you lost hydrogens, right? So oxidation is loss. So water was oxidized, right? Carbon dioxide, right? Hydrogens were gained. Reduction is gain. Carbon dioxide was reduced. So I could, again, on the exam, put a reaction like this and ask you which molecule is oxidized and which one is reduced, okay? So again, they always happen together. So if we look at cellular respiration, kind of overall, it's the opposite of photosynthesis, okay? So again, I'm putting this here to illustrate the oxidation and reduction, but also because these are what we're going to focus on in the next two chapters, right? And obviously major processes within biology, right, in cell biology. Okay, so if we look at this one here, we look at glucose, right, and oxygen here. So if we look at glucose to carbon dioxide, right, we see hydrogens are lost, right? So oxidation is lost, so glucose is oxidized, right? And oxygen was reduced, okay? Oxidation is lost, reduction is gain, oil rig, okay? So we need to be able to pick out from a reaction which one was oxidized and which one was reduced. And I'll always use examples like this where you can follow those hydrogens, okay? Because I think that's it's just a lot easier to conceptualize for us right now, okay? And also is relevant to biological reactions that are going to occur in the cell, and particularly in photosynthesis and cellular respiration, which is the next two topics, right? Okay, so if you go a little bit more and focus on photosynthesis and chloroplasts, right? I probably already said all of these things. We did just say this, right? It says hydrogen atoms are transferred from water to carbon dioxide as glucose forms. Carbon dioxide has been reduced and water oxidized, right? We just said that. Carbon dioxide reduced and water oxidized, okay? All right. And it says the energy is provided by solar energy. We've talked about that before. All right, so it says the reduction of carbon dioxide to form a mole of glucose stores 686 kcals in the chemical bonds of glucose, okay? And then you can oxidize that glucose in the mitochondria, right, to basically get that stored energy out. All right, so that leads us to mitochondria here, right? So again, you're going to oxidize those carbohydrates, break down those carbohydrates to generate energy, okay? I already said this by showing you basically those two reactions, okay? Glucose is oxidized, okay? And oxygen has been reduced if we look at that cellular respiration reaction, okay? Cells oxidize glucose step by step. So we'll talk about that when we talk about cellular respiration in more detail. All right. If we look at the cycle between photosynthesis and cellular respiration, and again, I think I kind of alluded to this in the beginning of the chapter with that sort of picture of the moose and the tree, right? But now we're focusing a little bit more on the cellular level and talking about the chloroplast and the mitochondria, okay? So it says carbohydrate produced in the chloroplast become fuel for cellular respiration in the mitochondria. Okay. Carbon dioxide released by the mitochondria becomes a substrate during photosynthesis in the chloroplast. Again, you can think about this cycle. Okay. Both organelles are involved in the redox cycle. Okay. Carbon dioxide is reduced in photosynthesis and carbohydrate is oxidized in cellular respiration. Okay. So again, we have this cycle. Okay. So it says energy does not cycle directly between those two organelles, right? It flows from the sun through each step of photosynthesis and cellular respiration. So again, complex. We're going to talk about these two processes in general, and we'll see how that sort of works out, right? So again, right, it's not this direct cycling through, okay? But you can see how they're related to one another, okay? So again, we think about a plant, photosynthesis. Photosynthesis occurs in the chloroplast of those plant cells. Okay, sunlight comes in. The plant cell is able to convert that sunlight into usable energy, uses that energy to make carbohydrates. Okay, then animals like the moose in that first picture will eat those carbohydrates, and then in those carbohydrates, those more complex carbohydrates get broken down, right, digested, and then once they're in the form of glucose in the mitochondria, they then get oxidized and broken down further via cellular respiration and basically generate ATP, okay? And you can see oxygen, right, is required for cellular respiration. In cellular respiration, CO2 and water are going to be given off. Carbon dioxide is required for the plants to be able to do photosynthesis and generate the carbohydrates. So again, I hope you can see the relationship between photosynthesis and cellular respiration, okay? And again, our next two chapters focus on these two processes in detail. Okay, so we will then be talking about them next. All right, thank you guys.